Introduction. The study titled Isolation of Tumor-Derived Extracellular Vesicles from the Plasma of Dogs Affected by Intracranial Tumors Showing Heterologous and Cross-Species Tropism, a Pilot Study, delves into the potential of using extracellular vesicles, or EVs, derived from canine brain tumors as a tool for both diagnosis and therapy. It begins by noting the similarities between brain tumors in dogs and humans, both in terms of incidence and prognosis. Malignant brain tumors lead to severe neurological issues and have poor outcomes, despite treatments like surgery, chemotherapy, and radiation. Unfortunately, these tumors often develop resistance to treatment, leading to recurrence. This has led researchers to seek new treatment strategies. Extracellular vesicles, which are nanoparticles secreted by cells, have emerged as promising candidates for drug delivery. These vesicles contain a variety of molecules, including nucleic acids and proteins, and are involved in cell-to-cell communication. They can also deliver cargo to other cells. The study highlights that tumor-derived EVs, specifically, have the ability to target tumor tissues, known as homing capacity. Previous studies have shown that EVs from both human and animal tumors can be loaded with imaging agents and that they selectively accumulate in tumor tissue. Furthermore, it has been demonstrated that canine glioma-derived EVs loaded with gadolinium can target murine glioblastoma cells. This research builds on these findings, aiming to explore the heterologous and cross-species tropism of canine patient-derived extracellular vesicles, or cPDEVs, specifically from brain tumors. The ultimate goal is to develop a safe and effective method to isolate and utilize cPDEVs for clinical applications, potentially as a theranostic tool for canine brain tumors, with implications for human medicine as well. The study aims to establish a sterile protocol for isolating these vesicles from canine patients and test their ability to target tumors in a mouse model. Study design. The methodology of the study is outlined in the Methods section, detailing the steps from case selection to data analysis. First, canine patients presenting at the VetsWies faculty, University of Bern, with neurological signs suggestive of intracranial disease were selected. These patients were diagnosed with intracranial neoplasia based on MRI scans. 4 such patients were included in the study group, while 4 healthy canine blood donors served as controls. A strictly sterile protocol was established for the isolation of canine extracellular vesicles, or cPDEVs. Blood samples were collected from both groups using sterile surgical techniques to ensure no contamination of the samples. The collected blood was processed through multiple steps of centrifugation to isolate the EVs, including an initial centrifugation to remove blood cells, followed by ultracentrifugation. The resulting pellet of EVs was then resuspended in a buffer solution. The sterility of the isolation protocol was verified by plating the final EV solution onto agar plates and monitoring for any microbial growth. To characterize the isolated vesicles, several analytical methods were employed. Nanoparticle tracking analysis, or NTA, was used to determine the size distribution and concentration of the EVs. Zeta potential analysis was performed to measure the surface charge of the EVs. Cryo-electron microscopy, or cryo-EM, was utilized to visualize the morphology of the EVs. Immunoblotting was used to detect the presence of specific exosomal markers, such as CD63, TSG101, and CD9. To test their tumor-targeting ability, the cPDEVs were loaded with indocyanine green, or ICG, a fluorescent dye. This was done by adding the EVs to an ICG solution and then washing away unbound dye. Murine models of subcutaneous tumors were created by injecting mice with colorectal cancer cells. These mice were then injected with either ICG-loaded cPDEVs, EVs from healthy donors loaded with ICG, or a solution of ICG without EVs. In vivo fluorescence imaging was performed 24 hours after injection to track the biodistribution of the fluorescent signal, followed by ex vivo imaging of extracted organs. Finally, the data collected, particularly on the fluorescent signal from the tumors and other organs, were statistically analyzed using a t-test or one-way ANOVA. The study was approved by the Italian Ministry of Research and University and regulated by a departmental panel of experts. Results. The results section details the data obtained from the experiments. 8 dogs were included in the study, 4 with confirmed intracranial neoplasia and 4 healthy controls. The mean age of the dogs with intracranial neoplasia was 7.3 years, with a mean body weight of 19.9 kilograms. The control dogs had a mean age of 6.3 years and a mean body weight of 30.0 kilograms. The dogs with intracranial neoplasia were diagnosed through MRI, with findings consistent with tumors such as lymphoma, meningioma, and glioma. One glioma diagnosis was histopathologically confirmed post-mortem. Table 1 summarizes the main characteristics of each canine patient, including their breed, age, body weight, and the main MRI findings. Figure 1 displays representative transverse MRI images of the brain of each patient in the study, showing the location and characteristics of the intracranial lesions. These images are detailed in the table and figure captions to clarify the type of tumor suspected and its location. Figure 1a displays an MRI of case 1, showing a focal lesion in the right frontal lobe consistent with a lymphoma. Figure 1b shows an MRI of case 2, revealing a broad lesion in the right frontal lobe consistent with a meningioma. Figures 1c and 1d display MRI images of cases 3 and 4, respectively, with lesions consistent with glioma. Figure 1e shows a control brain MRI with no evidence of tumor. Figure 2 shows the neuropathology of case 3, including a macroscopic view of the brain in Figure 2a and a microscopic view of the brain in Figure 2b, which highlights the key features of an oligodendroglioma. In addition, the study successfully isolated EVs from the blood of both patient and control dogs. The sterility test confirmed that the isolation protocol maintained aseptic conditions. Nanoparticle tracking analysis revealed that the cPDEVs from the patient group were larger than the EVs from the healthy control group. This difference was statistically significant. The mean diameter for cPDEVs was around 190 nanometers, while the mean diameter for control EVs was between 130 and 160 nanometers. Table 2 shows the full-size distribution profiles of EVs from each case and control, including the mean, mode, standard deviation, and percentile diameters. The EVs were found to express the typical exosomal markers TSG101, CD9, and CD63. The surface charge of the cPDEVs was more negative than the control EVs, as revealed through zeta potential analysis. Figure 3 contains data about the characteristics of the isolated EVs. Specifically, Figure 3a shows a representative particle size distribution of cPDEVs from case 1. Figure 3b shows the expression of TSG101 in cPDEVs. Figure 3c shows the surface charge of the cPDEVs and the healthy control EVs. Finally, Figure 3d shows the morphology and size of the EVs obtained through cryotransmission electron microscopy. Furthermore, the in vivo homing experiments in mice showed that ICG-loaded cPDEVs selectively accumulated in the subcutaneous tumors. Figure 4 highlights this finding, demonstrating the intense fluorescence signal localized only at the tumor site in mice injected with cPDEVs loaded with ICG. Figure 4a gives a schematic of the experiment, while Figure 4b shows the in vivo imaging of a representative mouse with the tumor, marked by a red arrow. Figure 4c shows the ex vivo images of the tumor and various organs, revealing the fluorescence at the tumor. Figure 4d is a quantification of this fluorescence. In mice injected with ICG alone or with ICG-loaded EVs from healthy control dogs, there was no specific accumulation of the fluorescent signal in the tumor, but there was some signal in the liver. The mean radiance released by the tumor in mice injected with the cPDEVs was significantly higher than the radiance from other tissues. These findings demonstrate that cPDEVs, derived from canine patients with brain tumors, display a heterologous and cross-species tumor tropism, meaning they are capable of targeting tumor cells of a different type colorectal and in a different species mouse. Discussion. The discussion section interprets the results and puts them into context of previous work and future directions. The study demonstrates, for the first time, the in vivo heterologous and cross-species tumor tropism of cPDEVs derived from canine patients with intracranial tumors. This builds upon earlier work that had shown the feasibility of isolating tumor-derived EVs from canine cell cultures. By loading cPDEVs with ICG and demonstrating their ability to target tumors in a mouse model, the researchers established a proof of principle for the use of canine EVs as a theranostic tool. The established protocol, which relies on ultracentrifugation, allows for the isolation of large quantities of EVs from relatively small blood samples while maintaining sterility. This is important for potential clinical applications. The study suggests that cPDEVs could be used not only for imaging purposes but also for the targeted delivery of anti-tumor drugs. The observation of heterologous tumor tropism implies that cPDEVs could also be used to identify tumors of different origins within the same animal, especially given the occurrence of multiple tumor types in older dogs. Since primary brain tumors are common in older dogs and can often be accompanied by other malignant tumors, this finding has potentially significant clinical implications. The study also addresses the limitations of the current research. It is noted that using a single ultracentrifugation step to isolate EVs may not be the most refined method for purifying specific EV populations. However, the authors chose to use this method to demonstrate the homing capability of EVs from tumor patient plasma to tumor tissue. It is further noted that the method is adequate for demonstrating homing capability, as homing was observed only in EVs from tumor patients, not from healthy donors. The small sample size of the study was acknowledged, and it was noted that this could affect the generalizability of the findings. The researchers note that, in future work, they aim to include a larger and more diverse sample of canine patients. The heterogeneity of the brain tumors included in the study was also identified as a limitation. It is recommended that future studies should aim to work with homogeneous tumor groups or stratify data by tumor type to fully explore and analyze the unique characteristics of each tumor. The absence of histological confirmation for every case is also addressed. The researchers note that brain biopsies are difficult for both economic and logistical reasons. They also cite previous studies that indicate that MRI is a reliable diagnostic tool for intracranial tumors in dogs, even without histological confirmation. The researchers emphasize that the results of the study remain valid and relevant. Overall, the study serves as an important step for future research into the potential therapeutic application of cPDEVs for canine intracranial tumors. The clinical relevance of the findings is also emphasized, noting that cPDEVs can be utilized as a tool for advanced preoperative and intraoperative tumor labeling. By loading cPDEVs with imaging agents like ICG, iohexol, and gadolinium, more precise tumor labeling can be achieved, potentially improving surgical outcomes. The researchers plan further studies to explore the full therapeutic potential of cPDEVs. Conclusion. In conclusion, this study demonstrates the potential of cPDEVs as a theranostic tool in veterinary oncology, with implications for translational research in human oncology. The ability of these vesicles to specifically target tumors, demonstrated in a cross-species model, could revolutionize the way canine cancers are diagnosed and treated. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.